When, and above all, how, did life evolve on our planet? This existential question, which we all ask ourselves at one time or another, animates scientific communities the world over. To answer it, paleontologists have no choice but to uncover fossils and study them at length in order to date them and make use of the extensive data collected. While certain periods in the history of our distant past are beginning to provide some clues, one in particular stands out from all the rest, so exceptional does it seem. We're going to take a very close look at one of the most famous and important eras in the history of the Earth, and of life in particular. This is the Cambrian period. This phase of history marks the appearance of the first ancestors of the plant world, but above all, represents the dawn of the Paleozoic era, with the appearance of living beings that still exist today. Transported more than 500 million years into the past, our journey through time takes us to a time when the land was rocky and desolate, but the oceans were inhabited by mind-boggling creatures. This period in Earth's history saw the sudden appearance of almost every major form of animal life in a short period that is thought to have lasted around 25 million years. Scientists call this explosion of life the Cambrian Explosion. This event, unique on the timescale, was of vital importance for the emergence and development of life as we know it today. Dear traveler, welcome. We are about to embark on a journey into an extremely distant past when life was in its infancy. Thanks to the study of numerous fossils, we'll be able to go back in time. In surprising environments, we'll meet strange, enigmatic creatures who, although they really existed, became extinct without a trace, or almost without a trace. But before we begin our temporal adventure, remember to like the video and subscribe to the channel so you don't miss a thing. Thank you. More than 3.9 billion years have passed since the formation of planet Earth. The Precambrian phase, which successively encompasses the Hadean, Archean, and Proterozoic eons, was marked by the formation of the oceans, the cooling of the Earth, and the solidification of its crust, followed by the formation of the continents Columbia and Rodinia. But it also witnessed the appearance of life in the form of bacteria. Then later, during the Neoproterozoic era, the appearance of the Ediacaran marine fauna, with archaic organisms, but above all, the bilaterian metazoans. The Cambrian thus follows this first long period of almost 4 billion years. This is the first era of a long series that makes up the Phanerozoic eon, which extends to the present day, itself divided into 4 subcategories. The first corresponds to the Paleozoic eon, or primary era, beginning with the Cambrian. The second subcategory is the Mesozoic, or secondary era. Then follows the Tertiary, and finally, the Quaternary eras, grouped together under the Cenozoic eon, which saw the appearance of the first primates and the evolution of many species as we know them today. But let's return to the period we're interested in, the starting point of the Paleozoic era, the Cambrian period. Spanning the period from 541 to 485 million years ago, this is the oldest of the 6 Paleozoic periods. What happened during these 56 million years that marked a decisive turning point, to the point of being defined as the starting point of a new and one of the greatest divisions of geological time, namely the Phanerozoic Eon? Let's start with the etymology of the word Cambrian. The term was introduced in 1935 by Adam Sedgwick, a British geologist considered one of the founders of modern geology. The name Cambrian is derived from Cambria, the Latin name for Wales. The name was chosen because this region of the world is home to numerous sites where stigmata of the period in question were first identified. Thanks to stratigraphy, the science of the Earth that studies the different geological layers, or strata, geologists have agreed to divide the Cambrian into 4 geological subdivisions, themselves accumulating 10 successive stages. Thus, the Cambrian period covers the Terranuvian from 541 to 521 million years ago, and Series 2 from 521 to 509 million years ago. The Terranuvian alone represents more than a third of the Cambrian in terms of duration. These first 2 eras are also known as the Lower Cambrian and the Georgian. They include the Fortunian stage as well as stages 2, 3, and 4. The base of the Cambrian is marked by the appearance of the Treptichnus pedum fossil record. These are complex fossilized burrows found in seabed sediments all over the world. Treptichnus pedum are thus the stigmata of animal activity on the seabed as they move and search for food. Unfortunately, no fossils of these animals were found in the sediment studied. The mystery remains, all the more so as the organisms responsible for these branching, sinuous burrows are far more complex than the animals of the Ediacara fauna that existed just before. Stages 2, 3, and 4 are marked respectively by the appearance of archaeocyaths, the first reef-building metazoans, and small shelly fossils, which are shell-like microfossils of metazoans represented by sponges, mollusks, annelids, lobopods, and other forms that don't seem to belong to any current phylum. Trilobites also appeared during this period. Then comes the Meolingian period, from 509 to 497 million years ago, which brings together 3 additional stages: the Woolian stage, the Drumian stage, and the Gainesian stage. During this period, trilobite varieties multiplied intensely. Traces of Erectocephalus indicus have been found in Guizhou province in China and in Nevada. Later came Titanokorys atavus, the now extinct trilobite species. Specimens have been found in western Utah. The base of the Gainesian 7th stage is defined by the appearance of the trilobite Lyjopyge levigata, discovered in northwest Hunan, China. Finally, the last Cambrian period stretches from 497 to 485 million years ago, and includes the Pabian, Zhangshanian, and finally the 10th stage, which forms the basis of the period that follows the Cambrian, the Ordovician. New trilobite species continue to appear during this period, but it is the appearance of the conodont, Lapitognathus fluctivagus, that marks the upper limit of the Cambrian. This prehistoric animal has the particularity of having cone-shaped teeth, as indicated by its name, composed of the Latin words teeth and cones. But what might the planet have looked like during the Cambrian period, which marks the beginning of the Lower Paleozoic? In the pre-Cambrian eon, Rodinia was a megacontinent that brought together all the continental masses. Under the influence of plate tectonics, it gradually fragmented. Pieces of continental crust began to drift away across the vastness of the oceans. Shortly before the Cambrian era, two new continents appeared. Laurentia, the larger of the two, forms much of the North America we know today, minus Florida, as well as Greenland and Scotland. The second, less important, corresponds to the Siberian Shield. At the beginning of the Cambrian, a third piece of Rodinia broke off. Named Baltica, it gave rise to present-day Russia, Poland, Scandinavia, and northern Germany. The amputated Rodinia continent was renamed Gondwana. However, it was still vast, encompassing South America, Africa, Australia, Antarctica, Southern Europe, and finally China. As the three newly formed landmasses of Laurentia, Siberia, and Baltica came into being, they were subjected to the effects of plate tectonics. They moved towards and away from each other. Oceans formed while others closed. The Lapitous Ocean opened up between Laurentia and Gondwana when the supercontinent Rodinia broke up. Over millions of years, this Paleo-ocean slowly expanded into the southern hemisphere, thanks to the spreading of the ocean floor in conjunction with a mid-ocean ridge located between the two continents. Around 50 million years after the opening of the Lapitous Ocean, at the end of the Cambrian, a new event gradually modified this part of the globe, reversing the opening process. A subduction zone south of Laurentia, where one oceanic lithospheric plate slipped beneath another, created an island volcanic arc. Volcanic discharges from the island arc mingled for a long time with the great thickness of sand and mud that had already accumulated between the Laurentia continent and this space riddled with volcanic islands. Sediments then accumulated in this zone, leading some 20 million years later in the Silurian to the closure of this ocean, all the more so as small continental masses, also uncoupled from the Great Gondwana continent, slid northwards. Together, these circumstances led to the gradual closure of Lapitous. As the insular volcanic arc moved closer to Laurentia, it eventually collided with the continental margin and began the long process of building a mountain range, the premise of the Caledonian orogeny, which would much later give rise to the Appalachians in North America and the ranges of Scotland and Scandinavia. The Cambrian began as the last ice age of the Proterozoic. The gas-gear's glaciation was coming to an end. A gradual and continuous warming of the climate led to average temperatures well above those we know today. But were the continents thus formed suitable for the development of any form of life? Although the climate seemed favorable, with average global temperatures estimated at 25 degrees Celsius or 77 degrees Fahrenheit, the continents that then occupied mainly equatorial zones were nevertheless, unlike the oceans, devoid of any signs of life. However, thanks to cyanobacteria, the first algae to appear and play a crucial role on Earth, the great oxygenation of the planet occurred, opening up the field of possibilities. Laurentia, Siberia, and Baltica thus enjoyed a tropical climate. Gondwana, located near the polar zone, experienced relatively cold temperatures, but without a land cover of ice. The intertropical zone, on the other hand, was more arid. The atmosphere contained 20% oxygen and 0.5% carbon dioxide, but the ozone layer was far too thin to protect the surface continents. Relatively exposed to ultraviolet radiation, they were unable to support any form of life. With no plant cover whatsoever, the continent surfaces were totally barren, desert-like and arid. Only the movement of tectonic plates and intense erosion eventually altered the appearance of these lands, which consisted of rocky peaks or barren, cracked plains. The shallow oceans, on the other hand, were able to take full advantage of these conditions to develop the life already present, thanks to their warm waters. At the beginning of the period, only unicellular, then multicellular organisms populated the submarine space. Prokaryotes, single-celled microorganisms with no nucleus or nuclear membrane, such as bacteria and archaea, were found. Then, in addition to the many cyanobacteria, came the eukaryotes, which had appeared during the Proterozoic eon. But we'll see later how climatic conditions played a major role in the evolution of these species over the 56 million years that make up the Cambrian period. Some 600 million years ago, towards the end of the Proterozoic eon, just before the Cambrian era, the Ediacaran fauna, composed of soft-bodied, leaf and tube-shaped organisms, developed for almost 56 million years before suddenly disappearing. The reasons for this sudden disappearance remain an unsolved mystery to this day. However, traces of this fauna have been found all over the world. It was made up of primitive representatives of younger groups, essentially the Cnidarian phylum, soft and medusoid corals, annelids, segmented worms, and arthropods. One of the few survivors of this short period is the jellyfish. But the event that followed was of such importance that it marked the beginning of a new era, the Cambrian. The Cambrian is the era that marks the beginning of fossiliferous times. Following the last ice age of the Proterozoic, the planet underwent a transmutation. Temperatures were much milder, and an unprecedented extension of the epicontinental seas allowed underwater life to develop at an unprecedented rate, but not only in terms of quantity. On a geological timescale, new anatomies appeared on such a scale that we speak of a Cambrian explosion. This was the first evolutionary event of its kind, marking a decisive turning point in the evolution of life on Earth. Some authors have even dubbed it the zoological Big Bang. Most of today's major metazoan phyla appeared between 541 and 505 million years ago. Although some of these multicellular animals have since disappeared, this Cambrian explosion was the initiator of a great diversification of animal, plant, and bacterial classes and species. Living organisms acquired shells, carapaces, and even skeletons, leaving their mark on fossils. Most of these were soft-bodied animals, such as worms and sponges, but the first arthropods, distant ancestors of crustaceans and insects, also appeared. There are also traces of the cephalopods of the future, including the octopus. The rapid emergence of new animal varieties, particularly bilaterians, made ecosystems much more complex than they had been before, when they were populated by relatively simple organisms, as witnessed by the Ediacaran population. Completely new ecological interactions, such as predation, appeared in new marine environments and habitats. Some so-called benthic species lived on the seabed, or even in the sediment covering it. Others, on the other hand, evolved in the aquatic environment. These are known as nectonic animals. The causes of the Cambrian explosion are still not really known. However, several hypotheses have been put forward, some of which are linked to one another. The first is based on an increase in dissolved oxygen in the oceans following the end of the last ice age. There may also have been an increase in the production of phosphate and nitrate ions, essentially for primary photosynthetic production, and enabling phytoplankton development and mineralization of hard parts, such as shells and sclerites. Changes in the food chain, inducing competition between prey and predators, may have favored co-evolutionary mechanisms between different species. As a result, new and vital defense strategies, such as carapaces, shells, and spines, would have developed, offering primordial protection against predators, themselves finely equipped with visual elements. And then there's the question of whether small genetic changes may have occurred accidentally during the embryonic development of certain species. You're probably wondering, and rightly so, about the evidence that has been found from this period. It is indeed legitimate to wonder how fossilized traces of these microorganisms, which moreover lived exclusively on the seabed, can be discovered. If it's hard enough to bring to light the fossilized remains of dinosaurs that appeared almost 300 million years later, what stigmata can be exploited? Fortunately, a large part of the Lapitous Ocean seabed has been preserved. As plate tectonics continually shaped the Earth's crust, surfaces in the ocean depths were raised to mountain slopes. This explosion of life left many fossils that can be found all over the globe. Traces of the Cambrian period can be found all over the world, on every continent. However, current knowledge of life during the Cambrian is based on the study of the Lager state, the storage sites still available, where paleontologists are continuing their research. The Cambrian is also well represented in France. Numerous fossils have been discovered in the southwestern Massif Central, more precisely in the Montagne Noire, but also in the Pyrenees, Normandy, the Cevennes, in small outcrops in the Pays de la Loire, and finally in the Ardennes. The first Cambrian fossils to be discovered in France date back to 1888. Trilobite debris was identified in the Montagne Noire, where the stratigraphy subsequently proved to be particularly representative of the Cambrian era. The Burgess Shale is one of the world's most remarkable storage sites. Discovered on August 30th, 1909, by paleontologist Charles Doolittle Walcott, they are located near Mount Burgess, high in the Canadian Rockies, in the heart of Yoho National Park. It's a black shale deposit where fossils, most of them around 505 million years old, bear witness to the end of the Cambrian Explosion. Shale is a type of sedimentary rock composed of clay and silt. Often very friable, these rocks reveal the fossilized remains of surprising, even disconcerting creatures. As we noted earlier, the mountains of the Canadian Rockies were once the seabed of the Lapitous Ocean. Sediments were deposited in the deep waters in the form of mud flows, capturing the animals present in this area. The scarcity of oxygen slowed their decomposition, so over time they were transformed into black, thread-like fossils. Skeletal elements from the Cambrian period are generally small in size. They come from small-shelled fauna. Different parts of mineralized skeletons are studied, such as sclerites, the calcareous part included in the tissues of certain corals, spicules, the calcareous or siliceous constituents, forming the skeleton of sponges, tubes, or shells. These elements probably belong to several different types of animal, but what is most remarkable about the Burgess fauna is the appearance of specific internal and external organs. At last, the animals were equipped with legs, visual attributes, a mouth, digestive system, antennae, and many other special features. To date, almost 150 species of animals, algae, and bacteria have been recorded in Walcott's quarry. Thousands of specimens are divided into several collections, enabling scientists to study the ecosystem as a whole. Quantitative statistics are now possible for the first time, thanks to the sheer quantity of fossils. Researchers estimate that almost 98% of the fossils on this site are those of soft-bodied organisms. The site's unique preservation conditions make it exceptional. Indeed, in most Cambrian marine deposits worldwide, this type of fossil is absent, not because they weren't there, but because they were not preserved in the right conditions. The Burgess community is essentially made up of animals. A few species of green algae and red algae have been recorded, as well as microbial colonies. But the groups most representative of this new environment are undoubtedly the arthropods, those small animals with articulated legs that molt, and the porifers, i.e. the sponges. Without further ado, let's take a look at the species that populated this paleo-ocean, then called Lapitous. Let's start with the most widespread animals, the benthics, i.e. those living in or near the seabed. Canidaspis perfecta is one of the ancestors of the arthropods found today in the form of crustaceans, insects, and arachnids. This specimen, which also left traces in China, measured from 8 to 52 millimeters in length. Its body was partially covered by a carapace and ended in a segmented tail with a telson at the tip, which itself had a double spiny projection pointing backwards. Its bivalve carapace was surmounted by 2 eyes, between which a spinal column began. It was equipped with 1 pair of short antennae and another, longer pair, whose function was probably olfactory. 2 pairs of legs were located at the level of its head, all equipped with a terminal claw. More than a dozen segments made up its body. The thoracic part was equipped with 8 pairs of associated biramous legs, similarly equipped with claws, while its posterior abdomen was devoid of them. Legs or appendages are said to be biramous when they have both gills and locomotor appendages. Canidaspis perfecta walked along the seabed, stirring up mud with its feet to find its food. Once the food was found, it used its pair of mandibles and maxillae to feed. Among the animals that inhabited the seabed, many species with elongated bodies resembling today's worms lived on the seabed or in the sediment that covered it. Others had a particular morphology that enabled them to swim actively. These elongated animals belong to the category of annelids. 5 species have been recorded in the Burgess Shale, including Paleozoic, which preferred a sedimentary environment, and Canadia spinosa, which swam with ocean currents. Let's take a look at the Canadia spinosa. This annelid worm measured an average of 3 centimeters or 1 inch long, and could reach up to 4.5 centimeters or 2 inches in length. The main characteristic of this animal is the numerous rigid bristles that extend from its dorsal branches all the way down its back. This feature is typical of polychaete worms. On their distinctive segmented structure, each segment carries a pair of conspicuous lateral appendages called parapodia, used primarily for locomotion. On these parapodia, tufts of a variable number of bristles, the chaetae, facilitate their movement in the aquatic environment. The Paleozoic, on the other hand, preferred contact with sediments. This tiny, thread-like hairworm takes its name from the Greek words paroni, meaning needle, and kate, meaning long hair, in reference to the bristles that cover it. This worm grew to between 1 and 2 centimeters or 1 inch in length, with a body just 1 millimeter wide without the bristles. Its body consisted of around 25 segments, each equipped with a pair of parapodia. This carnivorous, even detritivorous animal had a straight intestine running the length of its body. Its cephalic region was equipped with a pair of tentacles. This worm was endobenthic and epibenthic, which means it could move both on sediments and within them. Its bristles, very useful for its movements, probably helped it to bury itself in the sediment, both to hide and to search for food. But a much larger worm was discovered in the astonishing Burgess fauna. This is the Capinitator Praetermissus, a worm that proved to be a formidable predator during the Cambrian period. Considered a monster, it measured 11 centimeters or 4 inches, and its broad head was lined with 50 hooks. Nicknamed a Sagittarian worm because of its arrow-like shape, it is described as a chatognath. The name chatognate means jaw hair. In fact, it was its numerous movable hooks that enabled it to capture its prey. This worm is considered a branch of marine predators. Even today, this type of worm can be found in all seas. Although they haven't changed much, they are now often only a few millimeters long. Yet nestled among the plankton, it behaves like a fearsome predator, its hooks claiming numerous victims among the larvae of fish and crustaceans. The seabed was also occupied by brachiopods, animals that fed on suspended particles and whose bodies were housed in bivalve shells. Generally, they attached themselves to a surface on the seabed or to another larger organism by means of their peduncle, a cylindrical, flexible organ. Micromitra burgessensis was one of these brachiopods, whose maximum size was 10 millimeters. Its name comes from the Greek and means little turban. It was the most equipped of the brachiopods found at the Burgess site. It had a mineralized shell. This was adorned with pronounced striations of growth and fine raised lines raising out from the hinge across the entire shell, creating small diamond-shaped patterns on the top. Its valves were subcircular, while its hinge was practically straight. This specimen was also adorned with long, thin bristles that extended well beyond the margins of its shell. These were attached to the edge of the mantle, along the dorsal and ventral valves. Some of these specimens have been found fossilized while attached to the spicules of a variety of sponge, the Piranha sponge, confirming its benthic lifestyle. In the waters of this ocean, animals were actively swimming. This was particularly true of the chordates, animals with dorsal ropes and dorsal spinal cords. Pikaea gracilens, one of the fossils found in the Burgess Shale, undoubtedly represents the most primitive form of chordate. At the time, this ancestor of vertebrates only looked like a worm. However, it has been demonstrated that this animal was equipped with myomeres, the muscles of vertebrates. It measured around 5 centimeters or 2 inches in length. Its elongated body, with a head at the front, gave it the appearance of an eel. Its body, flattened at the sides, suggests that it had a ventral fin located towards the rear. Although this animal spent a lot of time on the seabed in search of food, looking for organic matter, such as snails or sea urchins, it could probably swim at will. One of the arthropods had 2 pedunculated eyes. This is the Sydneyea. With a maximum size of 160 millimeters or 6 inches, it had a convex, cephalic shield, short but wide and subrectangular in frontal view. The 2 lateral corners were notched to accommodate an antenna and eye stalk on either side. Its 2 hemispherical, highly reflective eyes pointed above and behind each antenna. Its thorax was composed of 9 segments, the middle ones being wider. The first 4 thoracic segments had appendages with a large base and 8 lighter ones ending in a sharp claw. The next 5 thoracic segments had identical appendages, but these were associated with branches of filaments, forming what could be described as a wing. Finally, Its abdomen was made up of 3 much thinner segments and ended in a triangular telson, accompanied by a pair of large articulated paddles, forming a caudal fan. This carnivore swam and walked on the seabed with the help of its 4 anterior pairs of thoracic appendages, while the filamentous branches of its 5 posterior pairs enabled it to propel itself through the water column. These same filaments may even have participated in the animal's breathing, acting as gills. Its spiny basal coxae helped it to crush food and transport it to its mouth. Its eyes and antennae helped it to move above the seabed in search of its prey, which it had to grasp and then crush with its front appendages. Another primitive arthropod had not 2 but 5 eyes. This was Opabinia regulus. Because of its unusual morphology, including a strange frontal proboscis, it is one of the iconic fossils of the Burgess fauna. Its body, whose maximum size was 10.1 centimeters or 4 inches, featured gills and lateral lobes repeated in series, as well as a generously proportioned caudal fan. Its complete body, measured without its proboscis, was between 4.3 and 7 centimeters, or 2 inches. On the anterior margin of its rounded head were 5 prominent eyes. They were bulbous in shape, surmounted by short peduncles. Its ringed proboscis was 4 times the size of its head. Extremely flexible, it ended in a pair of opposing pincers, each with 5 or 6 spines. The ventral mouth was directed backwards. Its trunk was composed of 15 segments, each housing a pair of lateral lobes, themselves bearing gill structures in the form of lamellae. The trunk was followed by a caudal fan with 3 pairs of upward-facing fins. This carnivorous animal swam thanks to the waves of undulations produced by its lateral lobes, which acted as propellers. Its caudal fan, like a rudder, helped it to steer through the water column. Its flexible proboscis and pair of distal pincers were invaluable for catching soft foods and bringing them to its ventral mouth. The seabed was certainly colored by echinoderms. Echinoderms are a distinct group of organisms characterized by a mineralized skeleton made up of several elements with pentameric symmetry. We find this structure today in starfish, whose bodies contain 5 identical parts, radiating from a central point. Naturally, carnivorous animals were fed on mollusks in large numbers. Of the 5 species discovered in the Burgess Shale, only one had a mineralized shell, the Scanella. Sponges also populated the ocean floor. They are among the most primitive animals known to the oceans. Their bodies had no real tissues. Many of these species had a skeleton made up of a mesh of fine mineral rods known as spicules. These demo sponges were benthic filter feeders. They therefore belong to the phylum Poriferae. Specimens of several species of the Voxia genus have been found in the Burgess Shale, including Voxia gracilenta. This sponge, very common on this site, measured a maximum of 8 centimeters. Simple and unbranched, or with more complex structures, each of its branches was almost cylindrical, forming a highly accentuated cone with a simple central cavity with a concave or flat opening. To feed, these sessile creatures had no other means than to continually filter water through the channels in their walls in order to retain the particles of organic matter then present. Other species, with variations in skeletal elements and branch shape, were also widespread, such as Voxia bullula and Voxia densa. These could reach up to 15 centimeters or 6 inches in height. Anchored to the seabed, they provided perfect shelter for epibenthic fauna, whose habitat was essentially the seabed. But the best-known sponge in the Burgess Shale is undoubtedly the Hazelia palmata. It is assigned to the upper group of demosponges of the order Monaxidina. The Hazelia genus is highly diversified. It includes species of various morphologies. Some were large and cup-shaped, such as Hazelia palmata, Hazelia crateria, and Hazelia luteria. Others were elongated and conical, such as Hazelia conferta, Hazelia grandis, and Hazelia obscura. There were also some particularly branched species, such as Hazelia delicutala, and others in the form of nodules or lobes, such as Hazelia lobata and Hazelia nodulifera. However, despite the considerable diversity of their general forms, these species all had the same microstructure. They were thin-walled and composed of small spicules densely grouped together in a mesh-like structure that opened outwards. Hazelia palmata, which measured up to 15 centimeters or 6 inches, was distinguished by its open, shield-like shape and numerous clusters of prominent radiating spicules. Among the arthropods active on the seabed alongside these sponges was the Naroya, often compared to trilobites. This marine animal, at most 4 centimeters or 2 inches long, had 2 ovoid dorsal shields with a convex axial region. It therefore had a cephalic shield and an elongated shield protecting the rest of its body. 2 long, articulated antennae protruded from the anterior shield. Behind the antennae were 4 pairs of cephalic appendages, with a further 14 pairs on the trunk. Each of these appendages was segmented and bi-branched. One part formed the locomotor branch, divided into 7 segments and thus perfectly adapted to walking. The other part was a filamentous branch, consisting of a fine shaft from which numerous lamellae sprang. These appendages ended in a large spiny plate. Thanks to its antennae, which were probably sensory, the Naroya was able to locate its food, which it brought to its mouth using the spiny plate at the base of its appendages. It could, however, take short swims, using its filamentous gills to propel it through the aquatic environment. If this carnivore was a scavenger, even a predator, the scars found on the carapace of certain specimens prove that it was also attacked by larger predators. Finally, a carnivore with a tubular carapace completes the list of arthropods. This is the Odaria alata. Most of this animal's body was enveloped by an imposing carapace, the midline of which was visible along the dorsal midline. These 2 united valves on the ventral part make this specimen very special, its carapace forming an open tube at the front and rear of the animal. Its head, which protruded from the tube, had a small anterior plate containing large spherical eyes surmounted by short peduncles. Its body consisted of around 47 segments, each with a pair of appendages. Those of the first two segments took the form of slender segmented branches adapted to walking. The other appendages were bibranched and divided into an inner branch with a large branched basal spine and an outer branch with filamentous lamellae. Its tail, or telson, consisted of 3 fins extending laterally and vertically. This predator's ventral appendages, blocked by its tubular carapace, probably made it difficult for it to walk on the seabed. Nevertheless, the Odaria was a mobile animal. It could plausibly swim in the water column, thanks to the undulation of its internal branches and biramid appendages. Like a suction pipe, it filtered aquatic organisms that entered at the front and exited at the rear of its carapace. Its large telson, very useful when on the move, helped it to steer, and why not, to manage rolling or braking when swimming. As you can see, this paleo-ocean was teeming with life, even if sea monsters were only small in size at the time. On a zoological scale, the Cambrian Explosion led to an unprecedented diversification of animal species. Above all, it marked the emergence of modern ecosystems. The biodiversity of marine life during the Cambrian is also perfectly illustrated in a region of southwest China. Various sites in Chengjiang, located in Yunnan province, are renowned for the quality of preservation of the fossils discovered, including their soft parts. The Chengjiang fauna came after the small shell fauna that marked the beginning of the Cambrian period. New life forms appeared at this time, some 520 million years ago. One of the sites, Maotianshan Hill, has been a UNESCO World Heritage Site since 2012. It was on this hill that this lager state was discovered by Dr. Qianguang Hao of the Nanjing Institute of Geology and Paleontology in 1984. The Chengjiang fossils comprise 16 phyla, over 170 genera, and more than 200 different species. They mark the first appearance of the majority of animal phyla existing during the Phanerozoic, many of which still populate our planet. The first specimens of algae, sponges, brachiopods, arthropods, chordates, and echinoderms have been found. But the long list doesn't end there. Hallucigenia is one of the most studied specimens. This animal, then considered a polychaete worm of the Canadia genus, has been extremely well preserved, providing experts with precious anatomical details. It had an oval-shaped head, attached to its cylindrical body by a constriction giving it a neck. Measuring 22 millimeters or 1 inch in length, it had 7 pairs of dorsal spines and 7 pairs of legs, with small claws at the ends. Today, it is classified as a lobopod. One of the most popular inhabitants of the Chengjiang fauna is a numerous arthropod, some specimens of which are in an excellent state of preservation. This is the Leoncholia illicabrosa. It can be recognized by its elongated segmented body, its biramid appendages, and above all, its 2 large head appendages, each bearing 3 flagellas about 2 centimeters or 1 inch long. Another common animal has revealed numerous anatomical details. This is Eurydichia intermedia, a trilobite that revealed fine uniramate antennae on the front of its head, large crescent-shaped eyes framing a rounded area extended by two long spines. Its posterior part ended in a long thorn-like tail. But the most famous fossil in the Chengjiang fauna is undoubtedly the Mylocunmingia vengioa. It represents the oldest known vertebrate. At around 2 centimeters or 1 inch in length, it is a small primitive fish. Its head was distinct from its trunk. It had 5 to 6 gills. Its trunk was equipped with a dorsal and ventral fin, probably twinned. The thorax had 25 V-shaped segments, with the tip turned backwards. While the location of its mouth is not yet clear, it did have a pharynx, as well as a digestive tract that extended to the tip of its tail. Another species stands out in the Chengjiang fauna, this time for its size. This is the Animalocaris sauron. This predator of the now extinct Animalocaridae family, also known as the strange shrimp, could measure over 1 meter in length, which is quite exceptional and even spectacular for this period of animal history. Its anterior part was equipped with a pair of large segmented prehensile appendages, edged with spines. Its head was surmounted by 2 large pedunculated eyes, while its underside featured a characteristic ringed mouth. Its segmented body, composed of 11 pairs of lobes, probably made it a very good swimmer. Thus equipped, it must have had no difficulty in feeding on trilobites, which it hunted with a sharp eye. In this complex ecosystem, scientists deduced that over 55% of species were arthropods, 10% were sponges. In addition, more than 10% are still of uncertain form. Most of the fauna lived on the ocean floor. Indeed, few of the specimens we have just observed were pelagic, as their structure was not equipped with elements that would enable them to swim, even passively. Feeding habits were equally varied. There were filter feeders and fearsome predators. While some of these animals were content to scavenge from the sediment, others were active hunters. At the Chengjiang site, the Cambrian Explosion is anything but a legend. The diversification of life is well represented, notably with the appearance of numerous phyla previously unheard of. While many descendants emerged within a single phylum, some endured while others died out in later geological times. Whatever the case, this period witnessed the emergence of all the complex forms of animal life as we know it today. In the lower Cambrian era, during the first 2 series of the Terranuvian and Series 2, a short period stood out, giving rise to the Tomotian fauna, named after the Siberian town of Tomot. This fauna appeared during the Cambrian Explosion, 530 million years ago, but lasted only a few million years. Yet, like the Ediacara fauna, it is similarly recognized around the globe. The Tomotian fauna is also often referred to as the small-shelled fauna, and for good reason. While it contains some organisms that look modern for their time, most of its representatives are formed of tiny blades, tubes, needles, caps, or domes. For paleontologists, the nature of this small-shelled fauna remains a mystery. Are they simply scattered fragments of the very first types of shells, still imperfectly formed by unrecognizable organisms? Or are they remnants of pieces of shell that covered previously known but unidentified animals whose construction improved over time? Or do these strange finds correspond to unique organisms that appeared early before spontaneously disappearing? Until recently, the Tomotian fauna was considered to be the first fauna with hard parts. But recent discoveries of organisms with mineralized skeletons have shown the opposite to be true, the latter being found in the stratified layers of the late Precambrian and early Cambrian. Nevertheless, the Tomotian fauna is also important for another reason. It marks the appearance of a group of organisms that most probably belong to the Porifera phylum, that of the sponges, the Archaeocyaths. Although this phylum disappeared at the end of the Cambrian, Archaeocyaths were the very first bioconstructors in association with calcimicrobes, calcifying microbial communities. They were able to generate organic masses measuring 1 to 2 meters in height and a few meters in diameter. These calcified sponges would then adorn the seabed like never before. These massive organisms were the precursors of the complex reef ecosystem that developed a few million years later, toward the end of the Ordovician period. All that remains of the Tomotian fauna is the sponge category. If organisms were able to develop on such a scale during the Cambrian, they were not the only ones. We have already observed a superpredator known as Titanokorys. This strange, extraordinarily large shrimp, sometimes more than a meter long, wasn't the only one to rain terror on the ocean floor. A recent discovery, published in June 2022, mentions the existence of another giant arthropod, belonging to the radiodont group. This is the Titanokorys gainesi. Its remains have just been found in the Burgess Shales fossil formation in British Columbia's Kootenay National Park. Its name, a reference to the word Titan, is self-explanatory and testifies to the exceptional size of this animal. The suffix korys means helmet in Greek. At around half a meter in length, the size of this animal was absolutely breathtaking at that time in animal history. While today it may seem almost insignificant, it's worth remembering that at the same period, the vast majority of living creatures were no more than a fifth of this size, with the exception of the Anomalocaris sauron we met earlier. Like its herded cousins, a subgroup of radiodonts, Titanokorys gainesi was characterized by an exceptionally long head, covered by a 3-part carapace of multiple shapes. The head of this specimen was so disproportionately elongated in relation to the rest of the body that some refer to it as a swimming head. Its mouth was shaped like a cone, with several radiating plates lined with teeth. Stemmed eyes with several facets crowned the mouth. A pair of long frontal appendages, ending in claws and pointing backwards, flanked its head. Finally, its trunk, made up of multiple flaps that were certainly intended to facilitate swimming, supported gills and ended in a front spine. This colossus, whose outer carapace was wider and thinner than that of the average radiodon, must have swum close to the seabed. Adapted to life close to the ground, it was most likely one of the most dangerous predators the oceans could count on during the Cambrian era. Smaller radiodonts, on the other hand, although with similar physical characteristics, were filter feeders. This probably explains the possible cohabitation of these two radiodont varieties, with Titanokorys gainesi certainly preferring to devour larger prey than its cousins. The study of exceptional faunas such as those of Chengjiang, Burgess, and many others, together with abundant paleontological data, allow us to assert that a complex marine ecosystem newly emerged at the beginning of the Cambrian. While the animal world preceding the Cambrian Explosion was already highly diversified, comprising several phyla each, with different strategies for exploiting the environment, the anatomical organization of specimens was still relatively simple. During the Cambrian, we witnessed not only an unprecedented proliferation of living organisms, but above all, the emergence of new anatomical and functional formations far more complex than ever before. From a sensory point of view, evolution is marked by the appearance of eye organs and antennae. Organisms are equipped with gills and a circulatory system, demonstrating that they had a respiratory system. In terms of feeding, complex digestive systems emerged. Some animals possess systems for capturing food particles, and in the case of predators, even prehensile organs. Finally, in terms of locomotion, more or less numerous appendages and flotation organs also appeared. Not to be forgotten are the prominent appendages found in arthropods, some of which have given rise to groups such as the crustaceans. These new anatomical peculiarities are accompanied by an increasingly complex marine ecosystem. Animals are no longer simple unicellular organisms like prokaryotes or eukaryotes, or even multicellular like certain myxobacteria. They are now endowed with a variety of specific organs, depending on which phylum they belong to. The discovery of organisms of different designs indicates the existence of an already highly structured food chain. Indeed, we find priapulan worms inside the sediment, numerous arthropods that lived on the seabed, and even swimming predators such as the fearsome Titanokorys. But the anatomy of these organisms, miraculously preserved in the fossils, isn't the only clue to learning more about these newly appeared animals. In some deposits, scientists have been able to analyze the stomach contents and coprolites of certain specimens. Observations of the fossilized food in the stomach, and even of the excrement still present in the intestines, has provided relatively precise indications of the diet of some of these animals. In this way, it has been possible to establish that certain species fed on other animal varieties. These stomach contents provide direct evidence of animal interactions within the food chain. In the Burgess Shale, several organic and skeletal elements have been found preserved inside the stomach or intestine of some of the animals studied. The priapulan worm, Otoya, seem to feed on hyoliths, those enigmatic horn-shelled organisms. Several aligned shells were identified in its digestive tract. The arthropod, Sydneia, on the other hand, had a varied diet. Numerous fragments of hyoliths, trilobites, and bradorids, small bivalve arthropods, were found in the digestive tract of this carnivorous predator, which measured between 5 and 16 centimeters, or 5 inches in length. But scientists weren't content to simply observe the digestive tracts of certain species. Among the fossils examined, some could be coprolites. These potential fossil excrements come in different shapes. Some, elliptical and composed of aggregates of skeletal elements, belong to the Chengjiang fauna. They turn out to contain mostly fragments of waptids and bradarids, the small bivalve arthropods that populated the seabed. These are probably excrements produced by one of the largest predators, the Animalocaris, which measured almost a meter in length. Other clusters discovered in the form of a ribbon and consisting almost entirely of hyoliths are undoubtedly the remains of carnivorous priapulan worms. These observations suggest that endobenthic predators, such as priapulans, were not the only ones to exploit the water-sediment interface. Arthropods, which moved along the bottom, and animalocarids, which moved through the water column, shared the same hunting grounds. In addition, traces of attacks and perforations were found on the carapace of some trilobites. Surely inflicted by animalocaridids, they testify to the bloodthirsty atmosphere that reigned on the seabed. Fractures and traces of regeneration, certainly due to predation, have also been noted on the shells of an Australian Lower Cambrian mollusk, Marocella. These small organisms, with biomineralized exoskeletons, were similarly subjected to constant predation. The Cambrian was thus marked by the appearance of the first ecosystems with a complex trophic structure, particularly in the animal sphere. Biological interactions, such as predator-prey relationships, are evident. New ecological niches, both benthic and pelagic, were occupied by a variety of predators, including scavengers, detritus feeders, and suspension feeders. Although the pelagic domain was probably not entirely occupied by swimming species during the Lower Cambrian, the concentration of animals in hyperbenthic environments close to the bottom is obvious. This was certainly the first step towards colonization of the water column, followed by the emergence of pelagic food chains. But what were the factors that triggered such an evolution? The establishment of this entirely new ecosystem is based on a number of decisive events and innovations. In the early stages of Metazoan evolution, climate, water, chemistry, and oxygen were most likely important environmental factors. But the acquisition by some species of a more complex nervous system, novel visual organs, and improved motor skills will certainly have enabled the introduction of new selective pressures such as predation. The light switch theory suggests that the introduction of vision into early Cambrian aquatic communities was the main trigger for this ecological renewal. This would have generated a completely new mode of animal interaction and triggered active predation. A cascade of feedback effects followed, such as defensive responses from certain prey, with the appearance of exoskeletons, biomineralization, and even new behaviors. Crucial improvements in the functioning of the nervous system in early Metazoans may have led to the emergence of new vital functions such as vision, limb coordination, locomotion, and even active predation. These new abilities undeniably modified the behavior of fauna, considerably changing ecological rules. Other hypotheses have suggested that major changes in phytoplankton composition took place during the transition from Precambrian to Cambrian. enabling an explosion in animal radiation. An increase in atmospheric oxygen could also have made possible the physical evolution of large animals with higher metabolisms. However, animal biomineralization during this same period is the most important event recorded. Its repercussions on the functioning of early ecosystems can only have been extremely significant. While the Cambrian period saw an unprecedented boom in the development of life, a first extinction followed the Cambrian Explosion, which seems to have peaked between 520 and 540 million years ago. But a significant event occurred around 485 million years ago. During this first period of the Paleozoic era, multicellular beings underwent one of the many biological crises that would affect all life forms in subsequent periods. Over 85% of the biosphere's species disappeared. So what happened on the planet to have such an impact on life? While it is acknowledged that 99% of the species that have ever existed on our planet eventually became extinct, the vast majority did so as part of a process of normal species extinction, linked to the limited duration of their biological existence. However, the Cambrian period saw the sudden disappearance of certain species, as did many others that punctuated the development of life on Earth. Some researchers suggest that at the beginning of the Middle Cambrian, the climate underwent rapid oscillations between hot and cold. These climatic changes would have been so rapid that many species would not have had time to adapt, and consequently to survive. A great global cooling and variations in sea level would have modified the terrestrial environment, considerably impacting living conditions on the planet. But what is their theory based on? Geoscientists have successfully dated lavas from the Kalkarindi volcanic province in Australia, as well as over 2 million square kilometers or 770,000 square miles of magmatic rock located in the Northern Territory of Western Australia. Their findings attest to the fact that these lava emissions, which occurred very rapidly, took place at the same time, around 510 million years ago. These events correspond well with the mass extinction of the early Middle Cambrian. Moreover, chemical analysis of these basalts shows that they are relatively low in sulfur dioxide. However, following a major volcanic eruption, an increase in atmospheric sulfur dioxide undeniably causes a temporary cooling of the climate. Could there have been a new period of glaciation, which, although less severe than the Veringer glaciation that ended the Proterozoic eon, would have decimated much of the nascent flora and fauna? Another hypothesis is that the oceans and atmosphere were depleted of oxygen and enriched with carbon dioxide. This global greenhouse effect would have caused global warming. But how did this greenhouse effect come about? Before the Cambrian explosion, the seabed was covered with organic matter that had accumulated over time, forming a thick, dense layer. According to some scientists, the rapid appearance of Cambrian fauna disturbed the tranquil equilibrium of the seabed. The animals, then in great numbers, would have stirred up all these organic layers. By fragmenting and decomposing the organic matter in the soil, the worms and arthropods triggered a mechanism that consumed oxygen and released carbon dioxide. As a result, this explosion of life became somewhat self-sabotaging, depleting oxygen not only from the oceans, but also from the atmosphere. As a result, the anoxic waters decimated the Cambrian fauna on a massive scale, leading to the extinction of numerous species of trilobites. However, geological records show that some species managed to survive, their bodies having adapted to this low oxygen environment. Finally, an extraterrestrial cause, such as a meteorite impact or the supernova explosion of a nearby star, producing a lethally powerful gamma-ray burst, could be the ultimate explanation. In just 10 seconds, the atmospheric ozone layer would have been destroyed, no longer protecting oceanic plankton from the devastating ultraviolet rays that were at the origin of the food chains then in place. A new ice age would then have slowly set in, covering the supercontinent Gondwana at the South Pole with an immense ice cap. With sea levels lowered and ocean circulation slowed, low oxygen levels would have led to much more difficult living conditions. The Cambrian era thus came to an end with a massive biological extinction, known as the Cambrian-Ordovician extinction. Brachiopods, conodonts, and trilobites were the animals that experienced the greatest reductions in species numbers. But as with every event of this kind, the surviving species take over the ecological niches left vacant. Nature shows unexpected resilience, and despite the unattractive environment, life continues to thrive. In a much calmer ocean, life returns to normal. Survivors multiply, and new species appear. At this precise moment in its evolution, planet Earth seems to be in full gestation. Will life, which first appeared in an aquatic environment, soon spread to dry land? If we can confirm that this will indeed be the case, will this spectacular evolution of species develop in the next period? For the moment, the Cambrian is giving way to the Ordovician period, a new era in which the global ocean and Earth's atmosphere cooled considerably.